Assalamu alaikum and welcome to Biocelia, where we will be looking at the small things in life because they make up the big things. In this first video, I would like to talk to you about plant growth promoting rhizobacteria. Now, PGPR are a very detailed topic, and we can't really go into perfect detail and describe every single type and every single element in it because there is a lot of genre and there's a lot of different plants. And to this day, we still don't know all of the patterns in which PGPR promotes plant growth. But for now, we do know the basic information that pertain to what makes a plant growth promoting rhizobacteria. So I'm going to try to make two videos. The first one is going to delve into the basic knowledge that you should know about PGPR before we dive into the second video, which will include the biochemical and the genetic elements that discuss PGPR. So let's get started. PGPR. The first thing we say is plants. So let's look at the plant structure. Now, a plant has leaves, it has stem and roots, which are integrated in the soil where we find microorganisms like bacteria and fungi, etc. The plant produces its nutrients by a process of photosynthesis. And produces carbohydrates by using CO2 and other elements and nutrients that come from the root system. Using these molecules, it builds structures like leaves and branches and flowers and fruits. Now, the plant has phytohormones, and at some time, the leaves are going to die, they're going to wilt, and they'll drop to the soil. In the soil, the microorganisms are going to degrade the leaves into organic matter. And due to a process of water and other elements, and maybe someone steps on this leaf and it is going to go back into the soil. It is going to be redigested, or at least these smaller molecules are going to be digested by the microorganisms into a more usable and absorbable form by the plant. So the plant is going to absorb these nutrients, going to repeat the cycle where it does a process of photosynthesis. It takes also water. By rain, it's going to use all of these elements, and the cycle is going to repeat itself. This is what we would call the biogeochemical cycle of a plant. In here, in the roots and the soil and the microorganisms, this is what we would call the rhizosphere. It is basically the space in which biological and biochemical interactions occur between the roots, the soil, and other microorganisms. So let's look at the soil. The soil is the space in which environments and other life that exists in here have certain interactions, and it is due to certain elements, like, for example, porosity, permeability. So, for example, porosity, it is the space between grains of soil that allow water and oxygen exchanges to happen and be contained. So basically, porosity refers to the ability of the grains to absorb these water and oxygen. Okay? Permeability, on the other hand, it is the flow of water and fluids into and through the grains. So what we say into, because even the grains, there are micropores that influence the bacteria life cycle. Now, there are certain elements which come into handy and basically are the interaction between soil and plant. For example, there's the nutrients and water acquisition. As organic matter flows and water flows into and through the soil, it is going to affect the drainage. It is going to affect the way that the root system is integrated into the soil and the nutrients that are absorbed, as well as a lot of the cycle that happens. Then there's the rhizosphere dynamics. For example, the roots, they are going to produce these substances and they are going to leave exudates in here. And there's so many, like, we can find enzymes, we can find certain sugars, we can find so many nutrients that are going to be used by the bacteria in the soil to degrade the organic matter and the nutrients, because it requires a lot of energy to do that. Then there's the structure of the soil, as we've said. In here, the grains, we can have big grains, we can have small grains, they can be different shapes, they can be formed of different substances, there can be different sizes. For example, there can be smaller and bigger, and this all influences the way that these transfers happen. And this is important to understand because the rhizosphere has certain factors that influence it, and we're going to look at them. So the rhizosphere has biotic and abiotic factors. Biotic meaning living, and in this example we have the plant, right? We have the organisms, the bacteria and fungi. However, abiotic, when we say elements, we mean the environmental and the physical elements and biochemical exchanges that are happening in here. So let's start with the abiotic elements because they are a lot easier to go through. So let's make this sort of table. One, we're gonna have the pH, we're gonna have nutrients, we have the temperature, and we have the water activity. And we're gonna look at how each one of these is going to influence the other. So we said that the plant cannot absorb certain nutrients in their form, in their current form, in the soil, and that bacteria are going to have to degrade it and turn it into a more absorbable form for the plant. In the soil, we have nitrogen, we have phosphorus, we have potassium, we have certain metals like iron, we have calcium, and these are all important. The nutrient nitrogen is going to be turned into either ammonia or ammonium in a process of nitrogen fixing by the bacteria. H4 plus. Potassium, we don't call it fixing or the fixation, we call it solubilization, because it is turned into dihydrogen phosphate ion. And this is far more soluble in organic format. Then we have potassium and calcium, which help in the ion exchange process and maintain ion homeostasis. Ion homeostasis by calcium and potassium. And there's other nutrients like sodium, stuff like that. They also help. Then we have the iron, and bacteria help the plant by chelating the iron so it can be used by the plant. It's a lot easier to find in the soil. Now, nutrients are going to affect the pH levels. Anywhere that we have a substrate, we have a concentration, right? So, for example, when the nitrogen and phosphorus are going to lower the pH level and make it more acidic. But in case for the calcium, now calcium is even used in agronomy to level out the pH and make it more alkaline, so it raises the pH levels. Wherever there's pH, there is water activity. And in this case, we're gonna be talking about water activity in regards to not only osmosis, but the availability of water. Let's look at the plant. So the plant is there's gonna be rain in the environment, and there's gonna be drainage. With certain high substances and different pH levels in the soil, and different grains, porosity, and permeability, what's going to happen is the soil is going to have different drainage levels, and it could be compact and it retains a lot more water. So in here, we're going to have higher water content. For example, if you take care of a plant and you overwater it, what you'll notice is that the plant is going to form droplets at the tips of the leaves or at the surface. And that's because the stomata in the plant cell, it is either going to open or close to allow photosynthesis. Now, when there's too much water, like a lot of the cells are going to shut down to prevent cell damage and loss of genetic material. By shutting down, it's going to be doing sort of osmotic regulation. And this osmotic regulation doesn't only exist in high water content, but also low water content. So if it's more sandy, the drainage is going to be really high. The plant's not going to find too much water. Or for example, in summer, and there could be a drought, the plant is going to shut down its stomata to prevent water loss in a process of aspiration. If the plant loses the water, there's going to be a certain loss of structure. So when it comes to water activity, high water activity, it is going to promote not only growth, plant growth, but it's also going to influence the strains of bacteria that exist. So it would be at water activity of 0.9 to promote bacteria proliferation, and at 0.8 for fungi. And there's also the humidity that comes into play on the surface, but we're not going to dive into this too much. So water activity, it can be high or low, and it's going to affect growth. Now we have the temperature. The water amount that exists in here and the stomata is going to influence and be influenced by the temperature. This is not necessary, like I just do this to show you a way to memorize each one. Now we have the sun and we have rain. Sun meaning high temp. We're gonna say low temp, and it's gonna be cold. We think of seasons like winter and stuff like that, right? When there's the sun, the plant is going to do photosynthesis. But if the temperatures keep on increasing, now the stomata is open, but if temperatures get too high, the stomata is going to be sort of confused. There's photons, but there's too much temperature. This could ruin the cellular structure and the hormones and the chemical reactions that occur. So stomata is shut down, and this will prevent water loss. But even if it shuts down, there's still water loss on a soil level. So with long amounts of time, there's a risk of a drought. So the plant has a sort of heat stress. And if the temperatures are low, then there's probably too much water. And in certain cases, as we've seen with the fungi and stuff like that, it could cause rot. And this rot is going to cause disease, phytopathogenesis. Like, for example, when the water droplets form in here, there's a lot more risk and diseases to happen on a like structure level when it comes to the leaves and the stems and the fruits, etc. So low temperatures. So we're also going to see a loss in water, and in here it's going to shut down. What's going to happen? It's not going to be doing photosynthesis. So it's going to lose nutrients. So as we can see, this is cold stress. And these two are going to result loss in water and loss in nutrients. But this, of course, like these differ on the cellular level, as well as different strains, different species, different plants, different bacteria, different soils, right? This is like a generalization of it, but it's not necessarily like the golden rule. I'm going to give you just a little note that we're going to go back to, is that in cases, let's do this, PS, in cases of cold stress, this can happen even in high temperatures, like in normal temperatures. Well, this can happen. Why? It's because there are certain microorganisms, certain bacteria, certain pathogens that can cause frost damage by certain biochemical reactions, and they're gonna cause the creation of ice and crystals along the roots, causing frost damage. Also, when we say heat stress, there's also heat damage, because on the cellular level, this is gonna cause heat damage. So heat stress, heat damage, cold stress, frost damage. And the frost damage can happen even in normal temperatures. And in all of this, you can see the close relationship between bacteria and the root system, right? This bacteria is fixed in nitrogen, it's solubilized in phosphorus, it's hydrolyzed iron. So you can see how this is an interaction that's happening. So let's look at all the possible interactions that can happen. So we're gonna look at, there's two. There's the direct and the indirect. As I'm explaining, I'm gonna try to give easier to understand examples, and then we're gonna return to how the plant plays into all of this. These interactions, they can either be positive or they can be negative, same as the indirect. For the direct positive, we have symbiosis. For the negative, we either have parasitism or predation. And while these two may seem the same, we're going to see a very small distinct difference that's important to distinguish between them. So let's look at symbiosis. Symbiosis, when we say it, we say survival. And it means that one bacteria is going to come into direct contact with another bacteria, or a bacteria comes into contact with the plant, right? And this bacteria is going to provide a certain nutrient or substance 1, and this bacteria is going to provide substance 2, and they need it for their survival. Either this one uses substance 2 for certain cellular structures and chemical reactions, metabolites, primary metabolites. We're talking primary, not secondary. And without it, it cannot survive. When we look at the bacteria coming into direct contact with the plant, the bacteria is doing nitrogen fixation, right? And the plant is doing photosynthesis. As photosynthesis happens, it's providing carbohydrates and sugars for the bacteria as nutrients, while the nitrogen is being used by the plant for its cellular structures and to do photosynthesis. So as you can see, the bacteria requires the plant to produce these substances for it to survive, and the plant requires the nitrogen or the more absorbable form of nitrogen for it to survive. So this is basically about survival. They need each other. It is a positive interaction. Now let's talk about the direct negative. We have parasitism and predation. And same thing, it's talking about survival. And when it comes to parasitism, it is when a parasite comes into contact with a host. Let's say a human, right? It's going to infect the human and it's going to use him for its survival. But the host doesn't necessarily need to die for the parasite to live and survive. So the parasite can live for a period of time in the human, finish its life cycle, and then either leave or die, or keep reproducing and then die, and then other parasites are going to use it. Host doesn't die for the survival. It's not obligatory. However, for predation, this bacteria is a predator. This one is the prey. And this predator needs to destroy him so that when it dies, it's going to provide it with the substances and the nutrients that it's going to use for its survival. So it is necessary that prey die for the survival of the predator. So for example, certain bacteria are going to target the membrane of the prey because it's the membrane that contains the nutrients, you see? This is the difference between predator and parasitism. Now let's look at the indirect reactions. There's distance, spatial distance between these bacteria. And this interaction is going to happen solely through the nutrients and the space. So let's look at the positives. We have cooperation. And we're going to see the difference between symbiosis and cooperation. So, for example, cooperation, we have two microorganisms in the space. For example, a biofilm or a petri dish. And one microorganism is producing substance A that is being used by this microorganism, this bacteria. And this other bacteria is producing substance 2 that's being used by this other one. They are beneficial to each other, but it is not obligatory. So it is not necessary and a requirement obligatory for them to be present for the survival of each other. However, in cases where certain substances that are being produced, antimicrobial substances, it turns from a cooperation into a mutualism. And I'm giving in a case of biofilm, but it's not necessary for it to be just in a biofilm. It is where one microorganism needs it to survive. So it is protecting this microorganism. And as it is protecting it, this one is producing more nutrients that are going to be used by this other bacteria. So this is just one case, but it can be so many other cases. And this means that it is obligatory for their survival to be together in the same space. And this is a sign of co-evolution. So we've seen cooperation, we've seen mutualism. Now let's look at commensalism. So for commensalism, it is basically when one organism is producing a substance that helps this one for like its main or secondary metabolites, and this one is not having any influence, like it's not producing anything that's beneficial for it. And it's not obligatory. Like, it's not a requirement for them to be together. It's just that one exists in the same space as the other one. So it's a positive neutral. Okay. We can also see this in certain biofilms, but I think the best way to like remember, especially these two, they're very important, like cooperation and mutualism. Now, let's look at the negative indirect reactions. Okay, so when we say, like, same as cooperation, when we say biofilm, we're gonna sometimes be talking about competition. I think it's easier to explain it outside of biofilm. So we have a petri dish. This bacteria is going to eat and grow in this direction. This bacteria is going to eat nutrients and grow in this direction. As they are growing and they are eating, they're going to meet at the center, the middle, wherever they meet, and there's gonna be a sort of competition over one, space, and two, nutrients. Okay, so in the soil, okay, we can see how these interactions come into play, because as these microorganisms are degrading certain nutrients, they are competing over nutrients and surface space of the root system. So when bacteria forms a biofilm over the root system and forms a sort of like a symbiosis, what's happening is that it is preventing other pathogens and other microorganisms and bacteria and stuff from adhering to the surface of the roots. So they are basically competing over the space of the root system and the nutrients, the exudates that are provided by the plant, as you can see. Now, another one is amensalism. Sorry if my writing is hard to read. So, for amensalism, it is where they exist in this environment and one microorganism or one bacteria produces an antimicrobial substance or a toxin that kills this bacteria, or it really prevents its growth or its biochemical reactions. Well, this one doesn't influence it at all. It is very neutral. So this one is basically negative, neutral, and this one is negative, negative. Another thing to note is that in case of predation, this interaction, we have parasitism, which is positive for the parasite, and it is negative for the host. And same with the prey predator, except that in the case of predation, it is good for the predator, but it is also good for a community of prey. So when it comes to a community of prey, the more prey there is being eaten and killed by the predator, the more space and nutrients are available to the community of prey. So it's basically positive for the community of prey and predator, but it is positive negative between the predator and the singular planktonic prey. I hope this makes sense. If not, you can just like ignore this. It's not necessary. This is the first bit, which is plant. I hope this made sense. This is the rhizosphere, and it is influenced by factors, biotic and abiotic. Abiotic, pH, nutrients, temperature, and water activity, and biotic as in interactions, direct, indirect, positive or negative. I hope this made sense. Now let's move into the growth. So where does the plant come from? It comes from a seed. This seed is going to germinate and then it's going to come out of its dormancy. As it is growing, it will be producing phytohormones for its growth. We're going to look at how these groups of phytohormones come into play with PGPRs. So phytohormones, there's a number of groups. There's auxins, there's gibberellins, cytokines, abscisic acid, ethylene, and there's so many more, but we're going to settle for these for now because these are the ones that we're gonna talk about most. So auxins, we're gonna look at an example, which is indole acetic acid, or what we call IAA. This phytohormone plays an important role in root growth. So let's say it's for the roots. Gibberellin, or let's just call it GA, is important mostly in the seed breaking out of dormancy and its process of seed germination. Let's look at abscisic acid. You're gonna find it mostly when the plant is in a state of stress. So it actually does the opposite. It induces dormancy. For example, another state of stress that we have already talked about is heat and cold stress. And what happens during these two phases is that the stomata opens and shuts to prevent water loss. So abscisic acid will also help with stomata function. Now, these three hormones are really important in the first stages of the seed. As it grows, there's going to be two directions. And this direction of growth is what we call tropism. The root grows into the soil, following gravity. We will call this gravitotropism. But as the stem and the leaves germinate and grow upward toward the light, we will call this phototropism. This reaction is going not only to be regulated by these phytohormones, but also directed by it, because prevent the top half from tilting towards the darkness. We've said that the auxin, for example, indole acetic acid, is mostly in the root. So what happens is that we will observe high amounts of indole acetic acid in the apical meristem in this spot. And as the amount grows, the cells are going to start to elongate downward and push the root cap to dig through the soil, growing longer and longer until it reaches a zone of maturation where root hairs are going to start to grow. This apical meristem is what we call the growth area or the growth zone, or it's basically that. And it determines the most important parts of the plant growth structures, which is what we call the apical dominance. This means that as the plant grows, it will mostly center its growth around the stem. But not only that, as the stem follows the light, now we have photons in here. So the plant has to follow these photons. So indole acetic acid will pile up in the opposite direction. And these cells are going to elongate. They're going to grow bigger and bigger, while this one is going to stay shorter. So what's going to happen is this plant is going to tilt toward the light. And same thing is going to happen if you put a source of light in this direction. Now, why is this important? It's important because the PGPRs, they are going to promote phytohormone productions, and it will be on different levels. So another one is going to be cytokinins. The plant is going to grow and it needs to do a lot of cell division, right? So cytokinins is going to help as well as prevent cell decay or leaf decay. Now, you think, but what is the thing that's causing leaf decay? Well, for one, it could be other bacteria, it could be pathogens, it could be some diseases, or it could be just a process of aging. And the hormone that causes aging is ethylene. And ethylene not only causes aging, but it also helps with fruiting and ripening. So when we see a plant lose its leaves and shed the leaves, it's going to be due to the ethylene. One thing that we would notice if we study phytopathology is that there's going to be some bacteria or some fungi that are going to stop the effect and degrade ethylene, or they are going to induce its reaction. So if a bacteria prevents aging, the fruit is not going to ripen. So when we see some fruits, they will just die before ripening, it's going to be due to these bacteria that prevent the production or degrade this phytohormone. However, in some cases, like for example PGPR, they will degrade ethylene into ammonia, and this will help them promote growth without stopping the process of aging but slowing it down. Okay, so as we've seen, is that water is going to be absorbed into the xylem up the plant, and it's going to cause either normal regular intakes of water, or it might cause a state of stress and an increase or too much water that will require the osmotic regulation thanks to the abscisic acid. But abscisic acid is also important in the process of germination and the breaking out of dormancy for the seed, for it to grow in a tropism, which is direction of growth either towards gravitotropism or light tropism, phototropism, and that will be also included in other elements like gibberellins, which will help it germinate and break its dormancy, as well as auxins that will help it know in which direction to grow. So as you can see, all of these phytohormones play an important role from the moment of germination to the growth of the plant. So I hope this was easy to understand. If there are any mistakes or something that I forgot to mention, then definitely leave a comment. Now we're gonna move on to the Rhizobacteria itself. Rhizo, I think we have already concluded that it means root. So they are the bacteria which exist on the root level. And the root level and that space is what we call the rhizosphere, right? The rhizosphere is the soil, the plant, and we have the plant surface as well. The soil is what we would call the exorhizosphere. The plant or the interior parts of the plant, which would be the apical meristem, the gland tissue, etc. These parts are what you would call the endorhizosphere. Now, the plant surface is the epidermis, and we call that the rhizoplane. Now, the reason why this is important to understand the differences between them is because this will define the type of microorganisms that we will find. Okay? Now, in the soil, the environment is a lot riskier because there can be pathogens, there can be insects, there can be protozoa, there can be so many different elements which will threaten the stability and the life of certain bacteria. Now, these bacteria are very specific. They prefer to be free living. That's what we call them. We call them free living bacteria. And for example, we can find some of them that will form spores that have certain mechanisms of protection, that can have certain capsules, that can have different structures. And for example, Azotobacter, this bacteria that we're going to take as an example, is a free living bacteria. It has so many flagella to help it move and adhere to certain substances and elements and stuff like that. It has between 30 to 40 flagella. It's a lot. It has also pili, fimbria, and these will help it with genetic and evolutionary and adherence and protection elements. These are really great nitrogen fixing bacteria. And the reason why I have Azotobacter in here is because we just need to kind of understand the structure of a bacteria and have kind of a reminder on how later on these elements are going to come into play. So this is basically just kind of like a reminder, but I wanted to use one that we're gonna actually learn about. Now for the rhizoplane, the bacteria that adhere to the surface of the plant, they are going to follow certain adherence mechanisms and then produce certain substances, which like EPS, if they form microcolonies, and then this will make up a big biofilm. And the biofilm is going to cover the entire surface and protect the plant from any pathogens that might try to infect the plant. So if we have like a pathogen over here, it wants to cause disease, maybe can cause frost damage, right? Some by stress, they can cause frost damage. So if it tries to stick and adhere to the roots and cause frost damage, these rhizoplane bacteria, which we will call epiphytic bacteria, and we're going to say bacteria with it because when we say epiphyte, we can also mean certain plants that will grow on the surface of bigger plants. So we have to be very specific and say epiphytic bacteria. They're going to adhere thanks to the pili, fimbria, the flagella, and then they're going to start producing and transcribing certain genetic material and producing substances, biochemical substances, cell wall, and they're going to kind of change in a form, while others are going to be more surface level. And when it comes to the biofilm, this is a biofilm. It has certain levels, okay? So for one, we have this as a support. The plant space is the support. Then we have the basal biofilm, basically the first bacteria that adhere. Then we have other structures like the surface. And then we have in here just the space, the soil. You can find so many different books or different ways in which they explain this, but as long as you understand that there is certain line where bacteria first adhere and then grow and attach and form microcolonies, then it's all good. So this is microcolony, and these microcolonies are gonna allow signals, communication, chemical exchanges, nutrient exchanges, genetic material exchange, where it also provides a tolerance to antimicrobial substances and antibiotics. And this is important. So it's kind of like a revision on how a biofilm happens and why it's important. So in here, as we've said, we have free living in the exorhizosphere, we have epiphytic bacteria in the rhizoplane, and then we have endophytic bacteria. Now these ones are mostly the ones that infect the cells, and it is through a sort of communication and signaling mechanisms between the plant and the bacteria. And if you kind of research online, you will find something called rhizophagy. And I would just like to just quickly note the rhizobium and the rhizophagy are not the same thing. And like, for example, rhizobium, they are the bacteria that would cause nodulation along the roots and in fact turn into bacteroids and then be a part in the nitrogen fixing cycle. Okay, but rhizophagy, it is when a plant phage, meaning eats a rhizobacteria. So this one is basically induced by the bacteria, even if there are communication with the plant, and the plant does help this process. But the rhizophagy is basically when the plant absorbs the bacteria. And the reason why I'm saying this is because based on my knowledge and my research, they require different genetic material, and this is the distinction between them. There's a lot of other different elements, but we're gonna focus on the rhizobium. So first, let's go back to the epiphytic bacteria. This has changed throughout the years, but in the original papers and books and stuff like that, you will find the epiphytic bacteria divided into types. Mostly today, when we say PGPR, we mean good PGPR. We mean good epiphytic bacteria. We mean ones that don't cause disease. But when we say epiphytic bacteria in general, we mean pathogenic, meaning ones that can latch onto the surface of the plant and cause disease. By the way, one other information I forgot to mention. Epiphytic, since it's on the surface of the plant, epi meaning surface and phytic meaning of the plant. So it would basically be on any part of the plant. Okay, it can be on the leaves, on the stem, and on the roots. But in this case, in the case of PGPR, we mean the rhizoplane along the root system. Okay, now the pathogenic ones, for example, we can say certain Erwinia strains. Like, for example, let's go back to the hormone groups. Certain Erwinia strains are going to influence ethylene levels, and they're going to prevent the ripening of fruits. The diseases that go a lot with strawberries sometimes will either cause rot disease or it can cause the falling of the fruit before the ripening. You see? So another type is biocontrol and bioinoculant. These ones can be used to defend the plant against the pathogen. Certain PGPRs are used as biocontrol and bioinoculants, for example. The bacteria that cause frost damage in regards to normal temperature levels. And that brings us to the third type, which is so interesting. It is called ice nucleation active bacteria. We're gonna call it INA. And this bacteria is going to produce ice nucleation proteins, INP, which will facilitate basically ice formation along the roots, as we've mentioned before. And this will happen at warm temperatures. But as we've said, in normal temperatures, and when this is happening, PGPR can be used as bioinoculant, which are cold tolerant, and they can protect the plant against these epiphytic bacteria. Isn't that incredible? Now, for the fourth type, it is the biofilm. which can be a lot of things. It can either be pathogenic or it can be used as a biocontrol and a bioinoculant. So I would advise you to go and see some videos on the formation of a biofilm. I think this is a very important thing to understand in microbiology. So it would really make a lot of things easier in regards to a lot of biochemical reactions that we will later discuss in the next video. So for now that we have understood what's the rhizosphere, the free-living bacteria, epiphytic bacteria, and the endophytic bacteria, I would like to give some examples. Okay, so this is something for us to kind of remember, or in case you have an exam or stuff like that, and you can reference these and know how to answer. So for example, you can understand that Azotobacter is a free-living bacteria. So Azotobacter. Now for the associative, we can find in certain papers extracellular PGPR or intracellular PGPR. You're gonna find them written in this pattern, but we're gonna call it associative bacteria PGPRs. One of them is very similar to Azotobacter. It is Azospirillum, but you can find so many other strains to remember. Another one is Clostridium. Now, I know that we spoke about Erwinia in regards to pathology, but guess what? There is also a PGPR strain types of Erwinia. So that's great. What else? Okay, another free living example. We have Pseudomonas. So we have Pseudomonas, we also have Bacillus. And now let's go to the endophytic ones. I really love the endophytics because they are so easy to memorize. And you'll see, I mean, we have Rhizobium, now we have Frankia, and last but not least, we have Brad. We have Bradyrhizobium. So this is basically it. I'm gonna check to see if I haven't forgot to mention anything. And yeah, I mean, this is nodulation. You can either check out some really cool videos, or I can include this in the next video. The structure, the rhizosphere. Oh yeah. So one thing I forgot to talk about is that a lot of bacteria are in the leguminous, but what's so interesting in certain cases of these bacteria that transform into bacteroids, PGPRs don't always turn into bacteroids, and it basically happens with non-galgoid plant species, which should be like warm season if I understand correctly. So the bacteria, once it infects the plant, it does not differentiate to a bacteroid. When it differentiates into a bacteroid, the genome is going to be amplified. And another thing is the cell is going to grow in size. It's going to have an enlargement. What's really interesting is that some bacteria, once they infect, they don't differentiate into bacteroids. And an example of a non-galgoid one, legume is soybean. So that's also interesting. One of the examples on bacteria, we have Bradyrhizobium. So yeah, I think this is a really interesting fact. If this is not that interesting, then you should just like ignore this. I just found this really interesting, but yeah, this is basically, I think this should be it. I hope you found this helpful. I know this is really kind of messy. My thought process is really messy and it's really intertwined, but this will all kind of tie down with the second video, and hopefully that's gonna be really soon. If there is anything that you didn't understand, please leave a comment below. And that's it. You should subscribe for the next video, so when it like comes out, you're gonna be notified. You also have to hit the notification bell, and that's it. I'm gonna try also to maybe later on at the end of everything, maybe make a really quick resume for people who are in a rush. And I perfectly understand that not a lot of people can just spend all their time really focusing on a 1 hour video or something like that. So bye.